Welcome to this class of analytical chemistry, where we will be discussing about the individual role of this particular subject. That means where we try to understand the role of analytical chemistry and the different techniques, because if we try to find out a particular technique which we can apply for any real life problem solving affair, that this particular technique should be compatible with that particular chemistry what can be utilized to solve this analytical process. So, there we get the different tools, basically the different techniques and the different types of chemicals that will be needed for a particular type of analysis. So, first thing or briefly we should discuss this particular role in terms of the sample, and already we are discussing about the corresponding sample and the sample preparation. From the real life examples what we have seen, that starting from the ore to deer kidneys, how we get the corresponding sample for this particular analysis. So, the different sizes, that means if we have the ore or any other thing, that we all know that the sizes are different. So, we choose that particular size, that size is important, and in which particular form those species are available, that we should take. So, this particular sample preparation should be associated with something which is known as treatment, or sometime it is pre-treatment. How we can utilize the knowledge of chemistry for the development of a particular assay, or the assay method we call, because these are the most tedious process, because to get this sample starting from say this sample is, if it is ore of very bigger sizes. So, we have to break them, we have to pulverize them, we have to make as the powder. So, during this particular process, definitely for the sample preparation we can find out this as the most time consuming step. So, most time consuming step is not that you get it in the final form and you determine by some titanometric method or some gravimetric method or some spectrophotometric method the typical unknown concentration. So, the sample preparation where the analytical chemist should intervene nicely for this corresponding preparation from the direct or the ore sample, and it is also the corresponding thing where we get that it is most error prone also. That means, if we are not careful enough to take the required precautions, so something can happen such that our desired form of the analyte coming from the raw sample is not possible to achieve it. And the third one is also, it is most labor intensive. That means, we must have to pay some time as well as some labor for that. So, it is a typical or a tedious process. That means, if we can have something, that means this ore, suppose we are handling something where you have the Fe3O4 and our idea should be there, we should use this, how we can convert this as Fe3 plus, which we can analyze or which we can determine the corresponding unknown concentration. So, this particular thing, that means, but if we can get this, there is a bigger chunk of these ores are there. So, bigger chunk of these ores. So, how to get this initially in the powder form and those powder forms you have to grind them and then you can take into some solution state. So, this Fe3 plus will be in solution. So, during this particular process, when we are looking for Fe3 plus only, but this particular ore sample can have other species present, and sometimes some valuable components are also present. So, during this particular process we should not lose any analyte for this particular analysis, because if some other metal ions, say Ni2 plus is there or chromium 3 plus is there, and if we focus our attention that we should only get this particular Fe3 plus nicely, but during this particular process, that means the dissolution, evaporation and all these, that some amount of Ni2 plus or some amount of chromium 3 plus can also be unavailable from that particular solution medium. So, once we get that and in which particular form, that means if Fe3 plus is present and we can directly determine Fe3 plus in the solution, that means the form of the analyte is also important. Also, sometimes that we add some reagent and the reagent can attack the particular species, that means Fe3 plus or AS3 plus or any other species, to form some colored species or some insoluble precipitate. So, this particular analysis, that means the analyte, what we get that. So, the form of this particular analyte is important for a typical assay process. So, if we get that a complex ore, and that complex ore, if we know that sometimes it is very complex, so we call it as the matrix. So, in that particular matrix, so this matrix we want to solubilize it such that we get it into the solution. So, there will be some interferences, and the interference present in the matrix can also make our life difficult to get these in the solution. And already if we have the interference present, so sometimes due to the treatment by the acid. So, if we have the ore matrix is being treated by the acid, so all these acids can have typical anions like chloride, like nitrate, like sulfate and all these. So, these were not originally present within this sample. So, due to that particular process, this addition of these acids will all be there, and this will be the new anions available in these solutions, because this particular oxide when treated by acid, it is not forming any anions over there, because all will be converted into water molecule. But if we treat this with some inorganic acid, so anions will be there, so they will also interfere. So, already we have some amount of interference present over there, and new interference can come over there due to the process of this sample preparation. So, what we see there, that this sample preparation can have something how we do the sampling process, the sampling. So, this sampling is also important when we take the powder sample for the analysis of iron, say of any ore, and this sampling is also important if we try to monitor a particular progress of the reaction. That means some conversion is taking place in some plant or in some other process, that a chemical reaction A is converting to B. So, the progress of the chemical reaction, so progress of the chemical reaction can be monitored, which is a typical challenge. So, sampling is a real challenge for any chemical analysis. So, if we try to do any chemical analysis, we should have the sample in our hand, and how to get these samples from any biological source, for any ore or any rock sample or any soil sample or any water sample or the air sample. So, this will further be complicated if we try to monitor the progress of a chemical reaction. So, this progress, what we see that this particular progress and how it is converting A to B. So, A is not directly converting to B. So, if there are some important intermediate species like X and Y. So, if these X and Y is forming, and those we are not able to monitor their concentration, but we are directly converting these to the corresponding final form of B, and this final form of B we try to find out by looking at the corresponding concentration of B which is forming from A. So, there are several things what can happen for this particular reaction. One is that if we can have a reaction flux, and within this particular reaction flux the reaction is taking place, and this particular reaction will not handle in such a way that A is directly converting to B without going through the intermediates X and Y. So, we can monitor a particular reaction in a flux in a lab. Then if there is a reactor in the industry, so or in a process plant in that particular industry. So, reactor is also responsible for any conversion, chemical conversion of A to B. So, we can monitor that, and in between, that means when the reaction is taking place for this particular process, suppose a metallurgical process, a furnace and all other reactions are taking place within that particular furnace or the reactor. So, intermittently or continuously we can monitor the progress of the reaction where A is not fully converting to B, or A along with X and along with Y, they all these are present in a mixture. So, typically A is not present, the entire concentration of A is not there. So, concentration of A is decreasing, but we are not able to get the right concentration of B, whatever amount of A is being lost. So, these intermediate concentrations are there. If we are unable to detect the concentration of X and Y, we will find that A is being destroyed, but is not happening over there. So, continuously we can also monitor the decrease in concentration of A and increase in concentration of B. So, this particular sampling process for this chemical analysis is also important on an open field. Suppose we are taking the river water, the river bed is our environment, and from there we try to get the water samples. So, from the river we can collect the water samples, but this is directly related to the environment. So, the location is very important, the time is also important, that when we are taking this particular sample throughout the year, throughout in the month or a particular day, in a particular time. So, where we are taking, when we are taking, and what we are taking along with the water sample. So, if it is contaminated with some species, we will be trying to look at that particular species from those river samples. So, sampling for the river is also very important. We have to go in the field, in the river bed, and we gather the corresponding river sample for the analysis. Then lastly, the other sample which we have seen the example in case of the death of the deers, that biological samples, so biological samples in a particular organism can be utilized the way we have seen in case of the kidneys of dead deers. So, this particular case, that means when we get the ore sample, so the nature of these samples are important, that means we are handling some solid sample, and this solid sample will be taken as the solution. And also we will see when we go for a gravimetric estimation, this particular solid sample, in between we convert it to a solution, again we go for some precipitation. So, solid we will be getting, and that solid can also be analyzed for the presence of that particular species, such as the ferric ion or any other metal ion in the original solid sample. So, what we do, we just get this particular solid, and one other important factor for the solid material of different sizes, the chunks and all these, if we are getting those as from the real life sample. So, these bigger chunks or the bigger size of this solid material are not homogeneous in nature. So, to bring homogeneity into this particular sample, how to make this particular homogeneous sample, because if we converted this particular solid sample of different sizes to a powdered sample. So, this powdered sample, making this as a powdered sample is important. So, we go for crushing. So, crushing is there. So, we crush that, then we can pulverize it, or finally we can grind them. So, to make this in the powder form, so how to get this powder basically. So, what are the instruments and what are the techniques we should follow to get a powdered sample from a very real life solid sample of different ores. That means, the physical characteristics of this particular solid sample is different from the sample which is in the powder form. So, we are changing the form. So, crushing, pulverizing and grinding. So, these are the three steps what we can apply on this, and we get this powder and then we mix it nicely. So, mixing of these, that means, if one part of the solid, so if we have one part of the solid and all these, because the geologists what find all these samples, so they are not homogeneous. So, inhomogeneity is there. So, when we are grinding, so one particular part, because these are all the crystallized form. So, one particular part can have some elements or some chemical elements of high concentration than the other part. So, when we grind this particular part, the concentration of one particular species is high over here than the concentration of this particular part. So, if we entirely make a powder and then the whole powder from this part and this part, we mix it, we get a mixed powder form, and this mixed powder form will be homogeneous in the solid state. So, what we will be looking for? We will be looking for something where we get homogeneous and which is mixed powder. So, these powder we can store basically from the different sites, like the collection of sample from the river bed, where from we are collecting that particular sample, which river, when, where and at what particular time. Similarly, the source of these solid samples of these ores are also important, and we make these, so we can have this powder samples. So, if we have 3 different samples from 3 different origin, so we make them powder and we can store it, because in the next step we have to set the corresponding process how we can analyze this powder form for their different analytes, the presence of these different analytes. So, before going for this grinding of this powder, so solids we can have. Now, we can bring 2 other forms, physical forms of these samples. That means, if we have this, that means during the sampling process, whether it is possible to go for the physical separations, because ultimately what we can do, that physical separations in sampling, we are still with that particular topic of sampling. So, in sampling the physical separations, because ultimately what we do is now known to us, is very much clear to us, is that we can go for some chemical treatment and chemically we try to identify the unknown species. But the physical separation, this can be very much useful if we can have all these things. That means, if we can have solid, if we can have the liquid, or if we can have gas. How these are there? Because if we can have one solid and if we try to separate from other solid, that means the separation. So, what is that? So, it is separation. So, separation of different components. So, if there are components A, component B and component C, and all of them are solid, then how to separate these components? So, separation of these components which are present within the sample due to the corresponding differences in the physical characteristics. That means, the physical differences are there. So, what about this? That means, if we have the difference, that means, if solid is mixed with liquid and liquid is mixed with gas, or gas is mixed with solid and gas is mixed with liquid, we can separate it out. But if there are some solid and we try to separate it from a solid, how to do that? So, separation of solid from a solid, that means, if we see that the different chunks are present and the color of those chunks and their corresponding nature is also different, that a particular scientist who are devoting with the geology or geophysics, so geologist can see the thing and sometime making this particular thing with some typical observation by microscopes and all. So, looking at this material, they can separate out some one particular part from the other, but in actual practice the science behind that particular separation is something else. If we just look at the density of one solid part from the other is different. That means, a particular part they are occurring in same area, some solid matter containing iron and some solid matter containing copper and some solid matter containing more precious metal ions, say like palladium or platinum. So, these are present in as the ores, but their density will be different. So, by density difference, using the corresponding parameter is the density difference. So, what we can do? We can do this separation by density flotation. So, what is that? So, density flotation is that if we choose some liquid and on that particular liquid the depending upon its corresponding density, whether it can float or it can submerge in that particular liquid, we can differentiate it out. So, if we have a particular density column where we have a density differences in the liquid, so using that particular thing we can use that thing like that of our liquid, but solid as and solid. So, solid versus solid separation, we just simply utilize the corresponding density of the solid material, the difference in the density of the solid material, or by solubility difference. So, one particular part is soluble in one particular solvent, other part is not. So, if there is any solubility difference, we just separate it out from a typical solubility difference for one solid from the other. So, next is that how we can separate a liquid from a solid also. So, a liquid is being, suppose it is being trapped, say a sample like calcium carbonate is in our hand, and that calcium carbonate has some trapped water. We all know that this we call it as the water of hydration. So, this trapped water we can take out. So, this as the corresponding liquid, that means the water vapor can be liquid from the solid of calcium carbonate. So, we simply heat the material. So, heat the material and trap the gas. So, this is the usual technique of getting this particular liquid form of water from the solid calcium carbonate. Now, if we have gas and if we want to separate gas from the solid material, what we can do that if there is a rock sample and this rock sample can have some gas, say we all know that the gas radon is trapped within some rock. Similarly, we all know nowadays that the corresponding gas available from the cell. So, cell gas we call, then we know that the methane is available from the coal bed. So, coal bed is trapping the methane gas. So, this is some particular physical technique is utilized to get that, but in this particular case if the rock is, if we get some bore of that rock and if the gas is coming out from those bores, we can collect those gases very nicely. Otherwise, we have to melt the solid. Once we melt the solid and then the gas is available, and then we purge that particular gas by some inert gas from there. So, next is that how we separate this solid from a liquid. So, the first thing is then the solid you have to separate from liquid is the filtration, the way we remove the precipitate from the solution. Suppose silver nitrate is added to a chloride solution. So, silver chloride is being precipitated as the precipitate, and if we filter it out from that aqueous medium, we get the corresponding solid as silver chloride from the aqueous medium, which is the water medium. Then liquid from the liquid, that means two liquids are there, and if they are miscible and if they are immiscible, we can have some thing which is called as distillation, because they will have the difference in boiling points. So, distillation is one particular technique, and sometime we find that if there are layers, so we can remove them by using say separating funnel we all know, which we can consider as decantation of the two immiscible liquid using a separating funnel also. Then gas from the liquid is also like that of our solid from the solid, then we have to purge then and trap technique. How we bubble some inert gas, say nitrogen or argon over there, and the gas what is coming out will be the gas which is we are looking for the separation, and that is if we cool those gases we will just get, if they are cold enough we can liquefy them, otherwise this gas can be separated from those inert gases. Or sometime the membrane, the gas permeable membranes can be used, which can selectively permit one gas to pass from the other gas. So, this membrane will be utilized for the separation of the gas from the liquid also, and the last one is the separation from a gas sample. So, what we are doing for the sampling process, we want to now separate the solid from the gas sample. So, again a different type of filtration technique will be used. So, solid we can have the suspended particles. So, suspended particles can be filtered out from the gas. So, the only thing that like that of our filter paper what we are using for separation of liquid, for the gas a different type of filtration mechanism we can use, such that gas can be used as the corresponding medium, where from the solid can be separated out. And similarly, from gas liquid can be separated out, such as our aerosol separation. So, liquid aerosol can be removed by filtration, again a typical filtration technique. So, this is a very useful one. So, we will discuss in detail also about how filtration is useful for all these different complicated types of separations. So, what we are looking over here is our separation of the components. So, we should not forget throughout this tabulation, that means what we are tabulating over here is the corresponding separations of all these things. So, filtration of the aerosol, and lastly the gas from the gas is by diffusion. So, the process what can be utilized is the diffusion technique, and this diffusion is different from one type of gas to the other, is the differential diffusion. So, this differential diffusion can be utilized the way we use it for a particular technique, instrumental technique. We know that this particular diffusion for gases is also possible. If we look at the corresponding chromatographic technique, we know that this is gas chromatographic technique. So, this gas chromatographic technique is the separation of these through some diffusion of these different gases, where we can have a carrier gas, and carrier gas is purging the other gases, and then mass wise the individual gas can be separated. So, if this is coupled with a mass spectrometer, the corresponding instrumental technique is the GCMS technique, and that GCMS technique is based on the diffusion of one particular gas from the other gas from which we want to separate this particular gas by this particular differential diffusion technique. So, this differential diffusion technique will be very much useful for separation of one gas from the other, because ultimately what we will be looking for, we will apply our knowledge of analytical chemistry to identify the separated gases. So, we just separate all these out, and then we apply this particular technique or one other particular technique to identify these gas from the gas where the gas mixtures were present. Okay. So, the next day we will just follow up these by utilizing of the different thing, that means the different techniques, that means the different tools and the chemicals if we can use for these particular techniques of analytical chemistry. Thank you very much.